Great. Okay. Hello, everyone. Thank you so much for coming. I am really pleased to be here today to help celebrate Olivia's accomplishments and for you all to learn a little bit about what she's been doing, which has been fantastic. I don't want to take too much time other than to say it's been a true honor to work with Olivia, both at an undergraduate level and then up through the TCBES program. You guys are in for a treat for what she's been doing. And I'd like to thank her committee members for being here as well. And with that, I'll present this with you, Olivia, and I'm excited to hear what you have to say, since I made this myself. Thank you so much. Okay, all right, there you go.
Hey, thank you all for being here. This is awesome. Yeah, most of you know who I am. I'm Olivia. I am a graduate student in the Tropical Conservation Biology and Environmental Science program. And I'll be presenting my thesis research on evaluating high-resolution remote sensing data and machine learning for detecting strawberry guava trees and its biocontrol on Hawaiʻi Island. And I'd first like to acknowledge all the people that made this project possible. First of all, my thesis committee, Dr. Ryan Croy, Travis Mendel, Elliot Parsons, and Jonathan Price, for all your feedback and expertise throughout the development of this project. Everyone at the Spatial Data Analysis and Visualization Lab, Pat for creating the object detection pipeline used in this project, Roberto and Tristan for teaching me how to fly drones and create flight plans, Sefa for all of his Lidar knowledge and advice in this project, Andrew, Kanoa, Alyssa, Ryan, and Sefa for all their help in the field collecting ground data for strawberry guava. David Okita was our helicopter pilot who helped us collect some of this imagery. Frontier Precision and Paul from Hawaii Community College allowed us to demo their LiDAR units and donate the data to explore for this project. I'd like to acknowledge Stacey Torigoy for inspiring strawberry guava to be the invasive species for this project. Tracy Johnson, who's here, for all of his knowledge on the biocontrol and efforts for tackling strawberry guava. All my friends and family that are here and on Zoom, for all their love and support throughout my time in graduate school. And everyone who's funded my time at Hilo, the Hawaii Data Science Institute, National Science Foundation, the TCBES program and geography department for having me as a teaching assistant, the Women's Cross Country program, and the STEM lab. A lot of people who helped me out. But I'm going to split this project or this talk into two parts. The first, I'll talk about the detection work for strawberry guava using aerial imagery, LiDAR, and machine learning. And then the second part, I'll talk about the detection work with the biocontrol using aerial imagery and machine learning. Let's start with strawberry guava. So strawberry guava is also known as 5E in Hawaii. It's the most abundant invasive species across the Hawaiian Islands. Strawberry guava is native to Brazil and was brought here in 1825 to be cultivated as a fruit tree. They're found most commonly in mesic and wet climates, in elevations ranging from 100 meters to 1,000 meters, and pigs are known for eating the fruits and spreading the seeds. These trees can grow to be 6 meters tall and form dense impenetrable thickets of skinny trunks like this picture and crowd out native vegetation. Because these thickets and how dominant this tree is in the forest, it's no longer feasible to completely eradicate this invasive, but it's still important to map its distribution to inform potential spreads to remaining native forests and conservation areas and inform biocontrol deployments happening on island. There has been recent work, very recent, a couple of years ago, done in Mauritius with Sentinel-2 satellite imagery, field data, and machine learning to map strawberry guava in Black River Gorges National Park, and that's what's outlined here. This was a really successful method for mapping strawberry guava distribution that would have been really ideal for Hawaii. with the large spatial extent satellite imagery covers. But strawberry guava in this study was the dominant canopy cover, whereas on our island, we have a lot of other trees in our canopy that are difficult to spectrally differentiate. And the image on the right is an example of a Sentinel-2 image of Ola'a forest. That's 10 meters resolution, and there's plenty of strawberry guava in this forest. And yeah, at the start of coming up with this project, yeah, I couldn't spectrally find strawberry guava here. And then on the other end, there was a study also done here in Hawaii in Puna that uses hyperspectral imagery and Lidar to assess gross primary productivity and above ground carbon density to identify areas of invasion. This is high resolution data that also performed well mapping strawberry guava here, but it's pretty expensive and complex to analyze. There are also maps estimating the distribution of strawberry guava across Hawaii based on elevation, vegetation class, and climatic conditions. It's no doing Kern. And through this project, I wanted to build off this previous work to explore remote sensing methods to map strawberry guava. I wanted to use slightly higher resolution than the satellite imagery so I can identify strawberry guava to start, but not hyperspectral imagery that would be too complex to learn for a master's thesis and find funding to use. And there's a lot of great detection work being done in the SDAV lab with drone and helicopter imagery and working with an object detection pipeline to process all of this data efficiently. So I thought this would be a great start to test this out for strawberry guava in our forests. So for example, this is an image taken with a camera attached to a drone. And where is the strawberry guava in this image? It's here all over, outlined in orange. And from an aerial view, the strawberry guava appears to be a lot more patchy than these circular tree crowns, and sometimes have less foliage than surrounding ohia trees. This is an example of an image taken during a period when the leaves are green, like the rest of the canopy. And this is the usual phenological stage of the leaves during majority of the year. But during periods of new growth in the springtime, strawberry guava leaves flush red and making it very identifiable compared to the rest of the canopy. This has been seen on the ground, but not so much investigated from the air. And this is an image taken from a helicopter. And it was quite the sight to see this in the air. And I thought it would be a feature that could improve detection. And so in addition to cameras and drones, another sensor we can attach to a drone is a Lidar sensor. And Lidar units emit pulses of light to measure the distance between the object it hits and the sensor to create a 3D model of the area. And this is an example of a cross-section of the resulting point cloud. And so the ground is brown and the rest is usually left unclassified in gray. And so this first part of the cross-section shows native ohia trees, lots of space in between each tree, and then next to it, we see a large cluster of strawberry guava trees. So I thought it would be interesting not only to investigate strawberry guava spectrally in the drone and helicopter imagery, but also to see if this structure was identifiable in the Lidar data. So with all of these types of data in mind, I investigate the following research questions. So first, how do phenological changes in strawberry guava trees impact detection success in high resolution RGB aerial imagery? Which forest structural metrics derived from Lidar data will be the most influential when detecting strawberry guava trees? And how do machine learning models trained with high resolution RGB aerial imagery or forest structural metrics derived from Lidar data perform when detecting strawberry guava trees? So let's start with this first question. So the study site for all the parts of this project is Ola'a Forest Reserve, and it's located on the island of Hawaii in the Moku of Puna in the Ahupua'a of Ola'a. And this forest reserve is a wet submontane forest that receives 3,000 to 6,000 millimeters of rain fall per year and ranges in elevation from 450 to 1,200 feet elevation, or meters, not feet. The forest canopy is dominated by native ohia trees and invasive strawberry guava trees, and then the understory is full of hapu'u and aluhe ferns. Smaller portion in blue of Ala'a Forest Reserve was chosen for this site due to the accessibility for the road, the presence of strawberry guava trees, and the location of the Forest Service SDEV Lab biocontrol project. And because strawberry guava typically grows between 100 and 1,000 meters elevation, this site is kind of an intermediate zone where strawberry guava isn't as dense as lower elevations, and it's sort of transitioning into a more native dominant forest. So this study site has a range of ohia forests scattered with strawberry guava individuals, like on the left-hand side, and then there's other parts like a couple meters down the road where you're dense with strawberry guava on the right. So due to the variability of strawberry guava at the site, it's challenging to find specific patterns throughout the site to detect strawberry guava, but this variability can also reveal interesting patterns across the gradient for detection, and it's investigated in this project. So the drone imagery was collected in June 2023 with a DJI Mavic 3 drone, and it's built-in camera, and the drone images were less than a centimeter resolution. A flight plan was created for the area in a lawnmower pattern, so lines going back and forth and overlapping. The images overlapped by 80% to stitch together and create this lovely image orthomosaic. The helicopter imagery for this section of the forest was collected in May 2024, so a year and a month later. The images were collected with Nikon D850 DSLR cameras that were mounted to a rig at the belly of the helicopter, and these images were just under 2 centimeters resolution. I cropped the images into 1,000 by 1,000 pixels and used this program called Label Image to draw bounding boxes of where strawberry guava was located in the images. And these bounding boxes, I made just over 5,000 of them on 747 images for the greenleaf phenology stage. And I also did the same for cropped helicopter images. Again, just over 500 or 5,000 annotations on 500 images. And our lab has a pipeline for detection model work for other species in the forest that I used for this project that's based on Deep Forest. And Deep Forest is an open source Python package that has an object detection convolutional neural network pre-trained on tree crowns from aerial RGB imagery that you can specialize to detect other tree species. And as a note, a neural network is just a type of machine learning architecture to process data and recognize patterns for detection. So the model takes in these images and these bounding boxes to help train it to identify the species. And I also implemented a 10-fold cross-validation, which means I created 10 models for each data set, the red and green phenology stage, and in a model, a different fold or subset of images and annotations are used to validate or evaluate the model's performance during training. So these are all the metrics I use to evaluate the model's performance. I calculate precision, which is the number of correct model predictions out of all the model predictions. Similar to that, recall is the number of correct predictions, but out of all of my annotations or bounding boxes. Those are also referred to as ground truths. And a box is considered correct based on a 40% intersection over union, which means if a model predicted a box that overlaps my ground truth box or my annotation by 40% or more, it's considered a correct detection or a true positive. I also calculate mean average precision by taking the area under the precision and recall curve, since this is a one-class detection where precision and recall are calculated at different confidence thresholds. An F1 score is the harmonic mean of precision and recall, and this is just another way to score model performance as balancing precision and recall. All of these metrics range from 0 to 1, 0 being poor performance and 1 is a perfect performance. And I also averaged these metrics across 10 models for cross-validation. And again, due to the variability of strawberry guava density and other trees present at the site, I did a lot of ground truthing to make sure what I was seeing in the data was actually strawberry guava trees. And the first few field days involved taking the GPS unit out and picking random clusters or individuals of trees just to get a sense of what strawberry guava look like in all the different ways. And those are indicated by these brown points kind of scattered throughout the road. And this usually involved a team of 3. So Panoa's in front there. He's making a path for us through the aluhe and a machete. Andrew was following behind with the GPS unit and would stand next to a lovely cluster for me. And I would record the point and any notes and pictures that were actually really helpful for looking back at the point clouds and the imagery. And in my most recent field days, I've created 3 20 by 20 meter plots that range from plot A, which was mostly dominated by ohia trees. Those are our blue points, and there are a couple strawberry guava individuals scattered throughout. Those are those light orange. Then we go to plot B, which has a mix of everything. We have ohia trees, clusters of strawberry guava trees, more individuals. And then I think there's a couple tropical ash trees in there. And then plot C is completely invaded by strawberry guava. So kind of all different increase in invasion across the plots. And I considered a cluster to be 4 or more strawberry guava trees together. And yeah, these plots were used to test the model and further investigate patterns, how the model performed as strawberry guava presence increased across the plots. And then I also created these polygons and digitized areas based on the ground truth points, the imagery and the Lidar data to better represent the area of the tree crown. Orange represents strawberry guava trees, and I kind of lumped everything else that's not a strawberry guava tree, which is mostly ohia in blue. So some results of these object detection models. I had a recall of 77% on the model trained on images taken during the green leaf phenology stage, and 84% for the model trained on images taken during the red leaf phenology stage. And in parentheses are the standard deviations, since I averaged the metrics, and the standard deviation is very low, which is great. That means the models are pretty consistent at predicting strawberry guava. And that means what I annotated here in orange, the ground truth boxes, the model also recognized as strawberry guava too in these blue boxes. And you can see a lot of overlap in my boxes and the model predicted boxes, which is pretty good. That means the model's learning and predicting what I'm telling it to recognize, even when it doesn't see my orange boxes. And for precision, I got a precision of 37% on the green leaf phenology stage model and 44% on the red leaf phenology model. And that means the boxes it predicted that didn't overlap with my annotations are predicted incorrectly, and those are considered false positives. And some patterns of false positives that I noticed were patches of ground, either it was grass, aluhe, dead trees that resemble kind of the branchiness of guava and the canopy. And then sometimes in the red phenology stage, a red omau fern would be predicted as strawberry guava. For the green leaf phenology stage model, I received an mean average precision of 79% and a mean average precision of 85% for the red leaf phenology model. And this graph shows the relationship between precision and recall at different prediction confidence thresholds. And again, getting low standard deviations. And all the results in a table. And with our final metric of F1 score that's balancing precision and recall, we see that the red phenology model is performing better across all the metrics, which is what we hypothesized, which is great. And then to check in on the model's performance on the ground truth plots, I had the model make predictions on images it hadn't seen before. So these images are roughly georeferenced to match the plots. And in this first plot, plot A, again, is dominated by ohia in blue and strawberry guava individuals. And there's one cluster in this plot outlined in orange. And so the top row, these red boxes in both images are the model's predictions. This top row is the green leaf phenology and the bottom is the red. So walking through these plots, plot A in green, these were the only incorrect predictions. They predicted on a eucalyptus tree just outside of the plot. And then the red leaf phenology model finds the only cluster in the whole plot, which is pretty cool. And then plot B has a mix of strawberry guava clusters and individuals, ohia trees, and other non-native trees. Both models had correct predicted boxes that capture this large cluster on the left-hand side of the plot and some other clusters, but it still misses a few trees not easily seen on the right-hand side of the plot. And that could be due to just it hugging an ohia and kind of being hidden. And then finally, the plot C, completely invaded by strawberry guava. Both models capture the dense clusters at the top of the plot, but miss a couple of individuals at the bottom. So across all the evaluation metrics, the red leaf phenology stage model performed better at detecting strawberry guava. For the ground truth plots, for the green phenology stage model, 34% individuals and 70% of the clusters were detected. And then for the red phenology stage, 17% of the individuals and 63% of the clusters were detected. And so there's significantly more clusters being found in the imagery than individuals. And that's, again, for more trees coming together. And so as strawberry guava invasion increases, the number of trees detected also increases in the plots. So on to the next question. Which four structural metrics derived from Lidar data will be the most influential when detecting strawberry guava trees? So switching over to Lidar, at the same study site outlined in blue, a Lidar scan was taken during October 2023 with the DJI Matrice 600 drone, and that carried this unit, the Voyager BiYellowScan sensor. And the flight I'm using for this project flew at 120 meters above ground at 5 meters per second and recorded a maximum of 8 returns. And that just means when a pulse of light is emitted, it reflected off of 8 different surfaces. And that's pretty good. Most Lidar sensors return 3 to 5 returns. And so this is just an example of the gradient. 3 to 5 3 returns is really good for getting canopy height and ground. And then as you get more maximum returns, you see more of the everything in between the tree trunks and understory. And so before I could create a model to detect strawberry guava from these data, some processing needed to happen. And I used the Lidar package in R to do that. And this involved filtering out any duplicate points and removing any outliers, like sometimes there were points hanging out way above the tree canopy in the red boxes. And I also normalized the height of the point cloud by subtracting the heights of the tallest objects in the point cloud. And that's known as the digital surface model, and then the ground, which is the digital terrain model. And instead of giving the model the raw point cloud to detect strawberry guava, I evaluated 4 forest structural metrics to see if any of these were indicators of strawberry guava. And this involved extracting these values from the point cloud into 1 by 1 meter raster cells. And the 4 structural metrics I derived and trained the model on include canopy height model. So again, subtracting the height of the trees from the ground, get canopy height. And then plant area index, which is the total plant area per unit of ground. And in this case, I did 1 by 1 meter cells, and it's calculated in this sort of 3-dimensional cell that's referred to as a voxel. And then foliage height diversity, which is the index of the distribution of plant material in the forest canopy. And so, for example, I would assume a strawberry guava tree would have a low foliage height diversity because there's not a lot of foliage throughout the tree. And then compared to a tropical ash tree where there's foliage throughout. And this metric also is used to measure diversity of vegetation throughout the different levels. So it'll capture any ferns you have in your understory. And then the final metric is point density from 4 to 6 meters. And the goal of this metric was to try and capture the high density of tree trunks and the clustering that occurs with these trees. So I thought that more points meant more clusters of tree trunks. And so I also digitized areas of strawberry guava in orange, again in other non-strawberry guava trees in blue, in this strip of road in the red rectangle. And this was done to assign a class to the pixels that represented strawberry guava trees and those that represented non-strawberry guava trees. And this strip of road was used for training and validation data. And again, I used the ground truth plots where I have recorded points to test the model. And instead of a CNN, I used a random forest, which I also generated in R for strawberry guava classification. And random forests are made up of several decision trees that vote for whether a pixel should be classified as strawberry guava based on the forest metrics values at that location. And the majority vote among several decision trees is the classification. And the model required 2 classes. So one was strawberry guava, and I lumped everything else into other non-strawberry guava trees. And so I used all the same evaluation metrics. And then I also added this one called mean decrease accuracy. And this just measures how much a model's accuracy would decrease if you removed it from the model. And this evaluates a metric's importance when classifying strawberry guava. And so the results in a quick little table, we have a precision of 0.79, a recall of 0.67, mean average precision of 0.72, and an F1 score of 0.72. And we'll come back to these. And then the importance of the forest structural metrics with mean decrease accuracy. We have plant area index is the leading metric that's most important for identifying strawberry guava with 45%. So if we removed plant area, the model accuracy would decrease by 45%, which is almost half, which is quite a bit. And then canopy height model is following in 42%, foliage height diversity with 34%, and point density in that 4 to 6 meter range where the trunks are is 27%. And to better interpret these relationships and results, let's look at the ground truth plots. So to set this up, for each of the ground truth plots, I tested the model's performance by giving those forest structural metric rasters for each of the plots to predict where strawberry guava is located here. I decided to visualize this by showing the ratio of votes for strawberry guava trees out of all the votes. And this is referred to in all the documentation as the probability. So we'll call it the probability of strawberry guava being detected. There are also raster cells missing from the plot. So if any of the forest structural metrics had a value of 0, I assumed that there was no tree there. It was likely ground. If it was canopy height is 0, or smaller shrubby vegetation. So in plot A, where ohia is dominant, there's lower probability values indicated in these blue to yellow pixels. And then in this red action with the eucalyptus, I think that is due to a lot of plant area. And then in this plot B, where there's a large strawberry guava cluster on this left-hand side, the probability is really high, which there's a large cluster there. And then we see slightly higher oranges and yellow probabilities in the other half of the plot. And there's less votes in the ohia trees. And then across plot C, lights up red, very high probability of strawberry guava there, which is very true because there is strawberry guava all over that plot. And this can also be visualized in the blocks plot. So across all the plots, the decision trees increasingly vote for strawberry guava classification as strawberry guava presence increases across the plots. And also looking at the breakdown of the forest structural metrics across the ground truth plots, these are cross sections of each of the 20 by 20 meter plots, and purple represents strawberry guava, and this yellow-green represents ohia trees, and the canopy height is smallest in plot A, where in the other two plots it's averaging around 10 meters. The foliage height diversity again is lower in plot A because there's a lot of individual trees, and then increases across the other two plots. And with the understory aluhe in plot B and C, I think that's also playing a role in this high foliage diversity, climbing up the tree trunks and hiding the stems of these clusters. And same for plant area index, shows the pattern as the density of strawberry guava increases across the plot, so does plant area, which we see a lot more purple as we move across the plots. And the metric I thought would represent strawberry guava most as invasion and clusters increase across the plots. And these values do match this hypothesis, but this band of 4 to 6 meters, aluhe is also sort of inflating these values. So when breaking down the forest metrics, it's difficult to make strong conclusions about which metrics are best for identifying strawberry guava at this site because of the variable densities of aluhe in the forest. Sometimes you have aluhe completely climbing up a tree, like in this red box, and then right next to it, there's no aluhe and you see the tree trunk just fine. I spent a lot of time working with a library of code called TreeSeg, and it's fit for filtering out the understory vegetation and extracting the tree trunks to try and rerun these models on just the trees, but the DBH or diameter at breast height was too small in the airborne Lidar to pick out all of the strawberry guava trees. But overall, the random forest had really great results from that earlier table. They were able to find patterns to correctly classify strawberry guava despite the aluhe. Plant area index was the structural metrics with the greatest importance to the model. And it was hard work, but it was really cool to see the airborne LiDAR, how it regenerated the forest, like ground truthing compared to the point cloud. It was pretty spot on and cool to work with. So now answering this last question and bringing the two methods together, how do machine learning models trained with high resolution aerial imagery or forest structural metrics derived from LiDAR perform when detecting strawberry guava trees? And here's the table with all of them together. And the LiDAR performed better, and random forest performed best in precision and F1 score, and the imagery performed best with recall and mean average precision. And overall, both remote sensing methods and models can detect strawberry guava, especially these clusters I'm finding in the plots. And the different types of data could improve detection. They work really well together. So scenarios where, again, that ground or patchy aluhe were misclassified as strawberry guava in the imagery, that's where LiDAR could help because canopy height is involved. And scenarios where ohia and skeletons of trees are falsely predicted in the imagery are areas where LiDAR could help because distribution of plant area and foliage height is also considered. And then on the flip side, when a model when the LiDAR incorrectly predicts a strawberry guava tree, the spectral information from the imagery could help out, especially during times of red flushing to detect strawberry guava. So by analyzing both of these methods separately, we can kind of break down what leads to a successful model, like phenological changes or differences in canopy height and plant area. Okay, that's it for guava for now. We'll jump over to biocontrol detection. I have some very fun photos. The Brazilian scale is also known as Tectococcus ovatus. It's been used as the biocontrol in forests on the Big Island since 2011 to reduce the spread of strawberry guava trees. The Brazilian scale is a very small insect that forms galls on the leaves or these swollen growths that feed on the leaf. And baby Brazilian scales emerge from these galls and like to target fresh new leaf growth at the top of the canopy. So these reddish leaves. Galling on the leaves can range from 1 to 2 galls to being completely covered in galls. And the biocontrol doesn't kill the tree, but reduces seed production and slows the growth of the tree. And Dr. Tracy Johnson and the Forest Service have done a lot of great work studying this biocontrol. And most recently, the SDAB lab has been helping to conduct releases of it and support monitoring efforts. And we do that by taking photos of the canopy every few months to see if it's working. So in the lab, I've done a lot of scanning through photos and zooming in to find galls on the trees. And when coming up with this project, I had read quite a few papers on machine learning models detecting different phases of plant disease, like here on the left with tomato leaves, and then also insect pest detection to assist monitoring pesticide treatments, like there on the right. And to my knowledge, I hadn't seen any machine learning work done like this for the Brazilian scale. Other papers mapped out the visual impacts of biocontrol with aerial imagery and machine learning, like some reduced flowering during a specific season or had wilted leaves due to biocontrol. So with all of this imagery that we have monitoring biocontrol in these trees, I decided to test out training another object detection model to identify gulls on strawberry guava leaves. The goal of this project was to contribute to biocontrol monitoring efforts and make the process of going through a lot of imagery a bit quicker, and just to see how well a machine learning model could identify the Brazilian scale, since it creates such a unique feature on these leaves. And so again, same study site used in the previous section. This is where the biocontrol was released 2 years ago, April 2023, in this L-shaped. And we return to these trees every 3 months, 6 months, and 12 months after deployment to take photos of the trees and monitor the biocontrol progress. These were the images I used to train my model, and also down the road to get some photos of trees where the galls have been established for several years. Yeah, down the road to get some of those photos. And I want to show a few examples of the imagery I used to train my model. This is a photo of a tree 6 months after deployment. And if we zoom into the galling, we see very minimal galling on the leaves. There's like 1 to 2 galls max. And then 6 months later, a year after deployment, this is a photo of a different tree, but same site. If we zoom in, there's a lot more spread of galls, and we attached biodegradable field tape to our initial drops to help us kind of identify where it dropped. So we're seeing some good spread after a year. And then this is an example of a tree where the Brazilian scale has been established for several years. And if we zoom in, these leaves are totally covered in galls. So it takes a while, but yeah. And not only do the images range in numbers of galls present, but also different heights above the tree. And so this shows an image a bit far away from the camera, causing the galls to look like this. And then another photo where we're a bit closer to the tree canopy. The galls are a bit more clear. So yeah, a wide range of variability in the images to see what galling looks like in the canopy. And I used the same program as before, label image, to annotate crop photos and draw these bounding boxes. I wanted to provide the model with some context for the galling. I didn't want to just draw boxes around the tiny galls that might be mistaken for branch nubs and different things, but also not the entire tree canopy, because that would kind of recreate the detection model I talked about before. And so as a result, the Brazilian scale was detected in every tree that had galls by the model, which is really great. And because I annotated at the leaf level, some leaves are missed, that's okay. My models averaged a recall of 80%, which means I annotated the leaves I annotated in orange. They were also recognized by the model in these boxes in blue. And with recall, again, we want to see a lot of overlap in these orange and blue boxes. And yeah, the model is learning again what I'm telling it to. I did receive a lower precision that averaged to 27%. And this means when the blue model predicted boxes did not overlap with my orange boxes, they are a bunch of false positives. And after looking through several images, some of the factors that I found to be false positives in this model were if we took images after a rain, The raindrops look like galls. During seasons of budding, the buds that are sitting right next to the leaf look like galling. And other things like blurry images with sunspots or any like discoloration of the leaf or a leaf transitioning from red to green. And just as a disclaimer, not all the trees that we deployed in developed galls, but over half of the trees did. And of those trees with galled leaves, biocontrol being detected in every tree is very exciting. Even if we miss some, that's fine. And this month marks 2 years since the initial deployment. So we hope to revisit these trees and put my model to the test again to see how the progress of the galling is going and to help the monitoring process. So in summary, mapping strawberry guava distribution is important to inform managers of new invasions and important conservation areas. These methods, the high resolution imagery in Lidar, can be a tool to detect strawberry guava in the forest, especially during periods of red flushing. Machine learning is an efficient way to process a lot of remotely sensed data to contribute to the mapping and monitoring of invasive species spread and biocontrol progress. And what's next for this work? Hawaii Volcanoes National Park with Stacy Torgoy is interested in mapping strawberry guava invasion that's creeping up Mauka in Ola'a Forest. And so Ola'a fence lines are here in orange, and this area they're interested in is highlighted in purple. And so we flew over the area with the helicopter. This is the image orthomosaic, all of them stitched together. And we flew about to the line where the invasion stops. And so, yeah, hope to keep training and testing out this model to support management decisions here, like in the park. And so that's it. Thank you all for being here today and supporting me throughout this journey of a thesis. And I can take any questions. Yes.
Well, hello, Olivia. Wonderful presentation. I probably missed it, but is there a specific month or season that the red flushing is happening?
I don't know the exact like day, but it's usually like end of April to early June, I believe. It's a very short window that it's happening. Yeah. So yeah. Yeah, and I think those images are taken in like the second week of May, so like right in the middle of it. Yeah. Yes, I see.
You mentioned that the IOU threshold was 40% for detecting the trees.
Yeah.
You didn't mention one for the leaves.
And it was also
It was also
40%.
40%.
Yeah. I kept the same model parameters for.
Is there a reason why it was kept at 40% not higher?
In the paper, the Deep Forest paper, they recommended 40% for tree detection and not higher. So I just stuck with the paper recommendation. But yeah, I think throughout making these models, I like played with every parameter you could. So that would be worth going back and seeing if making it higher is better. But yeah, I just went with the recommendation of 40%. Yes.
I've been interested in the Lidar methods for a long time. And one of the things that I think about is something that's so distinct about it is the density of stems and the fact that the stems are straight.
Yes.
So I wonder, in addition to density of points, there's a clever way to just count straight line points.
Yes, we tried thinking of ways we could do that, but I could not figure out. That's why I thought the tree seg, this like library of code, could like find straight lines of trees and bring that out for me. But it was looking for more of like a circle, like a DBH, but in the Lidar, it's very just, yeah, the straight line. So that's something we would love to do, and still thinking about, because that's like the feature of this we could get from Lidar. But yeah. Keely and then Natalie.
You said that like the model also accounted for like Velupe. Is there a way to like train it so that it knows that that's Velupe and not like, you know what I mean?
Yeah, I think if I was to switch detection from making these rasters and giving it the raw point cloud, there's ways to like trace or classify this like chunk of points of the Luhe, but sometimes the Luhe is like so far up the tree, I'm missing the tree. But yeah, that's definitely something worth looking at too. Natalie.
So if you were to do the model in a forest of different canopy structure, like one by, for example,
Yeah.
what do you think would be the standout metrics from those four that you ran? Think it would change because the canopy is so much higher there?
Yeah, I think that's something I wanted to explore in this project, was also looking at if it were in the understory, but I kind of took that out. But I think I think canopy height would still be significant in plant area because of these, like, we went to La Puebla and there's a lot of like just big clusters rather than these individuals. But yeah, I think still plant area. I think the value, so I wouldn't be able to use this training data for La Puebla, but probably recreate a new model. But I still think I did look at cross sections where there is a eucalyptus, a guava, and a luhe, and there is this like midsection where maybe exploring like this canopy foliage would be helpful and unique to guava. Yeah, I think it's still possible, but I don't know exactly what that metric would be yet. But yeah. Noah.
So Olivia, awesome job. You got some really helpful feedback about your precision with the galls, like, you know, having water drops on the leaves and stuff that
Yeah.
were triggering false positives. But other than Uluge, what was, did you like go back through those false positives and figure out like really big indicators or things that were affecting your precision just detecting guava itself? And could one of those things be the galls themselves? Like, are they showing up at that kind of scale?
That's a good question. I did look back through the images. I wasn't specifically looking for galls, and I don't think I saw any specifically, but maybe as more galls spread, that'll be a feature to look for. And then other things affecting precision for guava, a lot of like dead ohia trees were kind of popping up since it's more branchy. Also, like sometimes the camera wouldn't be looking straight down, it's kind of looking at a side. So like even a base of a eucalyptus or an ohia, it would just like capture, cut off at the canopy, but it would get the like bottom stem of it, I think, because it is such a branchy, I don't know the right word for it, but it's that feature it's picking up on. But yeah, I would say dead trees and the bases of trees are also leading to those false positives for guava.
Are you able to take those false positives and then validate them and then throw them back into the model to further increase precision? Oh,
Yes, yeah, that's something I could do.
awesome.
Yeah, and I think, yeah, that's, yeah, Roberta's currently doing that with myconia detection. Yeah, so maybe that's in the future for the guava stuff. Are there any questions on Zoom or? All good. Okay, we're right at 4 o'clock. Any other questions?
Okay, well, let's thank Olivia. all so much for coming, and please take some food, and we should get out of the library because I want to step down. What's that? Yeah, we can hang out a little bit.
Yeah, they know we're here.
They know we're here.
Yeah.